Okay? So you see the three different models that Messelson and Stahl were looking at. Messelson and Stahl. Okay? So out of these three models, they experimented and they tested and they did this whole series of experiments. And what they came up with was that basically DNA will replicate in this semi-conservative fashion. It's going to contain or keep intact part of that parent strand and make an exact, make a replica of that parent using this as its template, using it as a baseboard to start off and build the second strand with. Okay? So let's look at that, the semi-conservative model of replication. It starts off with a parent strand. This parent strand has complementary base pairing, adenine to thymine and cytosine to guanine. So we have this complementary base pairing every time in our parent strand. We can see they no longer, we're not even representing that sugar phosphate backbone. It's this outside parts of our ladder. Okay? And we're just concerned about those nitrogen bases in the middle running up and down. Okay? So we're going to have very specific very specific complementary binding, adenine to thymine and guanine to cytosine at all times. Well, in this semi-conservative fashion, what happens is these DNA molecules are actually unzipped, unwound. Those hydrogen bonds that are very stable in very large quantities are still very weak bonds and can break apart very easily. And so when DNA goes to replicate, it's like unzipping a zipper. We're just breaking those hydrogen bonds apart. And now we're separating it into two individual DNA strands. Still that parent strand. If this was my parent strand to begin with, okay, what I'm doing is I'm separating it into two parents. I'm just splitting it in half. I'm splitting it in half. And now what will happen is I will start adding my complementary base pairs. Because adenine will always still bind to thymine. Okay, adenine to thymine. In this daughter strand, adenine is still going to bind to thymine. And this strand over here, the thymine, is still going to bind to adenine. And it will form its complementary base pairs all the way down. And by doing that, you can see we are creating two daughter strands that are identical, that are still identical. If I do this again, okay, if this is my parent, I'm going to separate this out, right down the middle basically. Unzip those hydrogen bonds. And now what I can do is I will form exact copies. Don't you love my models? So high tech. Okay? I'm making, I need Lego, something. I've made exact copies now of that DNA strand. And it's simply because these complementary base pairs, adenine will always bind to thymine. So if it's adenine on my parent strand or thymine on my parent strand, I'm going to complementary base pair the other one. And I will end up with two daughter strands that are identical to what I had started off with. Two daughter strands over here, identical to what I started off with over here. This semi-conservative model of replication keeps part of that strand intact to give it as a building block, to use it as a model to create the new daughter strand. And what will happen the next generation, this will split apart again and use it as an exact copy all over again. Okay? This is the way that DNA replicates in a semi-conservative fashion. Semi-conservative fashion. Conservative would be making an exact copy. Yeah. Yeah, just literally making a photocopy of it. but there's no little Xerox machines inside of our cells. So we use part of it to make the other part.
Where did the new complementary base come from?
Good question. Where do the new complementary nucleotides come from? These ones that are filling in. Well, they come from a couple different places. First of all, we can make them inside of our nucleus with the proteins and different molecules. But more likely than not, these nucleotides are coming from foods that we digest, foods that we consume. We utilize carbohydrates for respiration. We utilize fats for cell membranes and insulation. We utilize proteins to make new proteins, or utilize amino acids to make new proteins. Well, in all of our food sources that we consume, we are still digesting DNA, also these nucleotides. And so we have nucleosidase enzymes that break them down also into the nucleotides that are brought into the nucleus to be utilized to create the new DNA strand. So really, we can generate them, build them inside, but we also get them, those nucleotides, simply from the foods that we consume. Okay? Any other questions on that so far? Okay? So we have this semi-conservative form of replication where we're splitting this DNA molecule in half, unzipping those hydrogen bonds, and allowing new DNA molecules to form complementary to it. This is what's happening during that synthesis phase of mitosis, or of interphase. This is, when we replicated DNA during the synthesis phase. That's what we're talking about here, is how that DNA is replicated. Now, we have 46 chromosomes that have to replicate. And those 46 chromosomes, some of them are really long, like chromosomes 1, 2, 3. They're very long chromosomes, like hundreds of thousands of nucleotides long. Our DNA altogether is about 6 billion nucleotides. All right, so there's a lot of nucleotides in our DNA. And when we go to replicate it, what we need to do is, or what happens is we don't start at one end and just slowly work our way to the other end, because that would take us forever. And interphase is a long period, but we don't have forever to do that. What happens when our DNA goes to replicate is that it starts to replicate all over that entire strand of DNA. If you've got this double-strand piece of DNA, all over the place are what we call replication bubbles, or origins of replication, where this DNA will start to split apart. Think of a belt that you put like a little bubble in if you fold it over and you try and, you know what I'm talking about? How you kind of bubble out like that, or a piece of string or something. DNA will do these replication bubbles. They will create these little blurps all along our linear DNA, all the way along it. And in doing that, it will start to replicate at hundreds of different places along that entire strand of DNA, not just at one end going all the way to the other end. It'll start all over in order for it to be completed very quickly. Does that make sense? Kind of, sort of? So if we look here, we can see, for example, this dark blue is our parent strand of DNA. The dark blue is our parent strand. And there's these origins of replication. This is showing 3 origins of replication, where it just kind of bubbles out. And DNA will replicate in both directions. It'll go, towards the left and towards the right on that top parent strand, and it'll also go towards the left and towards the right on the bottom strand. And well, here's a second replication origin, and these two will run to left and to right. Okay? And eventually, the two will come together, won't they? They'll run into each other. The DNA is replicating in this way, and the DNA is replicating in this way. They're eventually going to meet. And the more replication bubbles you've got, the more ground that's covered in the shorter amount of time. And so, in a matter of, a short time, you're going to have 2 completed strands identical to what the parent strand was. Does that make sense? Okay? So we're going to see this replication all over that nucleotide, or that nucleic acid, all over that entire strand. It'll just open up all over the place and start replicating. Okay? Questions so far? Okay, well, let's look first at how these nucleotides actually will attach in this complementary base pair fashion, building this new nucleotide strand. What we've got going on here is, here is our template strand. This is that dark blue piece of DNA. That's our parent strand. Okay? We have our parent strand. Now we need to bring in those complementary base pairs according to what this parent strand is telling us. So you can see here's the sugar phosphate, sugar phosphate, sugar phosphate going all the way down. Here are our nucleotides going all the way down. Well, if this is a thymine, then our nucleotide that we bring in is an adenine. A guanine will bring in a cytosine. A cytosine will bring in a guanine. Well, here's an adenine. What we need to do is bring in a thymine. The way these nucleotides are found in our nucleus or in our cytosol, brought into the nucleus and stuff, is going to be as a triphosphate. Well, we've talked about triphosphates, haven't we? Adenosine triphosphate. It's an adenine nucleotide. It actually has a ribose sugar, not a deoxyribose sugar that we have in the DNA. But it is an adenine nucleotide that have 3 phosphate groups. We have guanosine triphosphate, cytosine triphosphate, and thymosine triphosphate. These are found as a nucleotide, the nitrogen base, that sugar group, and then 3 phosphate groups instead of only 1. Well, we already know that ATP with those 3 phosphate groups are carrying a tremendous amount of energy, right? Allowing for this exchange of energy, allowing for things to happen. Well, that's what's going on with these nucleoside triphosphates. We've got all these phosphate groups there. What we're going to see is this chemical reaction, this release of some of that energy. Here's our sugar group, okay? On this sugar, and I'm going to kind of just write over here, we've got 1, 2, 3, 4, 5 carbons. Oh, well, that's not a carbon there. That's an oxygen. There's number 5. 5 carbons there. Okay, everybody see that? We've got 5 carbons on that sugar group. Well, this number 3 carbon here has our hydroxyl group sticking off of it, that OH. And if we look down here, this is number 1, 2, 3, 4, 5. Well, number 3 has that hydroxyl group sticking off down there on the bottom. Right? What happens are these phosphate groups, a phosphate group, remember, has oxygen, 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 basically hydrogens here, and then figure this is another phosphate, oxygen, phosphate, oxygen, oxygen, oxygen. Okay? Actually, I'm going to put the double bonded oxygen on the bottom.
Is the number 5 carbon double bonded to the phosphorus and the other carbon?
No. The number 5 carbon is still single bonded. It has hydrogens on it, though. Organic chemistry, we don't show those hydrogens. Okay? So basically what's going to happen here is we've got this phosphate group. I guess this is attached to the sugar. Okay? We've got 3 phosphate groups attached to our sugar here. Okay? Ready to react. Well, what happens is the hydroxyl group on our sugar and the oxygen, well, that's not right. The hydrogen on my oxygen. Yeah, that makes sense. Trying to form water, because remember, it's a polymerization reaction. We're joining the 2 together. We're forming water. The hydrogen on this phosphate group, because these are all hydrogens here, we're going to lose these 2 phosphate groups here. The hydrogen here and the OH group on our sugar will be removed as water. That'll open up this bond here on this carbon for this oxygen to bond to it. Everybody with me on that? Okay. And so what happens is this phosphate group here is going to react with this carbon here. We're going to remove water, which they're not showing. We're going to lose these two phosphate groups, which they are showing here. And we're going to have this phosphodiester, this diester linkage between the sugar here and the sugar here with the phosphate in the middle. Phosphodiester linkage. This thymine is guided up to the adenine by that attraction of hydrogen bonds, right? We talked about those hydrogen bonds. It's guided up to that adenine by those hydrogen bonds. And these phosphate groups here react, exchange that chemical, lose that energy, release this hydroxyl group here with the hydrogen over here to form this covalent bond. And now we have that next nucleotide attached with this hydroxyl group on this number 3 carbon waiting for the cytosine or the guanine to come in and bind with this cytosine, the next one up. The hydrogen off of this phosphate group here on this nucleoside triphosphate. Right. These two phosphate groups get removed. This is our thymine. Okay. It's a phosphate group. Right. It takes this hydrogen off and makes this covalent bond here with the next sugar. Okay? That's what's going on there. So it takes the hydroxyl group off of this sugar. The two of them form water, and the oxygen is now bonded. So now I have sugar, phosphate, sugar again. And then here's another hydroxyl group sitting here with my next phosphate group that'll come in and take this away and join again and again and again. Okay? Does everybody see that? How we form this sugar phosphate backbone now over on this side by that next nucleotide coming in, the next nucleotide, the next nucleotide, the next nucleotide. Okay, that hydroxyl group on the number 3 carbon, and this phosphate group is actually here attached to the number 5 carbon, isn't it? Okay, and this phosphate group, this circle here, was attached to this number 5 carbon, and so on. Okay? Everybody see what's going on here? Okay. Well, we're getting one step closer to understanding this. Now, I was so lovely to point out these carbons that were numbered, and now here we're showing the carbon up on top that's numbered. Okay, here's our sugar group. It's a deoxyribose sugar. This number 2 carbon is missing that hydroxyl group. That's what makes it deoxyribose. Flashback to chapter 5 if you don't remember that. The number 1 carbon is attached to our nitrogen base. Here's our number 2 carbon. Our number 3 carbon is what has that hydroxyl group on it. The number 4 carbon and the number 5 carbon is sticking out of the sugar with the phosphate group attached. Okay, everybody have a feel of where we are. Okay, this is kind of just repeating what we were just doing. Well, when DNA replicates, these DNA strands have to, first of all, run in opposite directions. Up and down escalator, north and south lanes, okay? Opposite directions of each other in order for those nitrogen bases to come in contact with each other in the middle. Another thing, these numbered carbons are very important because what we see here is this phosphate group is what we call now the 5' end. This is like maybe, for example, our first nucleotide. It has just one phosphate group there. It doesn't have the other two for a chemical reaction to happen with the next nucleotide. Okay? And you can see down here we have this, the number 3 carbon is what we call the 3' hydroxyl end, because that's where that hydroxyl group is. Okay, if we put this back up here real quick, I'll just kind of see if I can stick this down. Something like that. Okay. So this hydroxyl group here is on this 3' end. This number 3 carbon is where we see that hydroxyl group. And the phosphate group is up here on the number 5 carbon. Everybody with me on how this is flowing so far? The other side has to go in the opposite direction, doesn't it? I have to have my number 3 carbon up here and my number 5 carbon down here in order to get those nitrogen bases together in the middle. Okay?
What was the name of the hydroxyl group called?
The phosphate group. This one? Hydroxyl, 3' end. 3'. Yeah, that little tick mark at the top of the 3 is a prime mark. And so they just call it the 3' end or the 5' end. Okay?
So wherever the 5' end would be for the phosphate.
Right. The 5' end will have the phosphate group, and the 3' end will have the hydroxyl group. Okay? Now, when DNA replicates, new nucleotides will be attached only from the 5' to the 3' end, meaning it will only grow from, I'm going to attach the 5' phosphate group to the number 3' sugar. Does that make sense? It will only go in this 5', the 3' direction. I am growing from here to here, from 5' to 3'. And you can see how I'm starting up here and I'm just continuously growing this way. That 5' end is that new nucleotide coming in. That new triphosphate coming in has that number 5 phosphate sticking out on the number 5 carbon of my sugar. Okay? And I'm going to attach it to the 3' sugar of my growing chain. So I'm going from 5 here, the number 5 carbon here with the phosphate is going to attach to the number 3 carbon on the sugar already growing.
So one will grow up and one will grow down.
Exactly. This one, if we were to split this chain apart, okay, this template right here, split it apart, this side is exactly like this side, isn't it? Okay? It has the hydroxyl group over here, and my phosphate group is here on the number 5 carbon. So this chain is exactly like this parent chain over here. And now, because you see this is a 3' end to 5' end, the only way I can grow is starting at the top, going from 5' down to 3'. This side, if I separated it out, will grow from here up to here. Okay? It will only and can only attach nucleotides from the number 5 carbon of that triphosphate to the number 3 carbon of our sugar already in that growing chain. It has to. And so in turn, if I'm separating this out, if this is my 5' end here and my 3' end here, and I separate this out, I'm going to have to grow from here up to my fingertips, and then I'll grow from my fingertips down to my elbow on this side. So it goes like this. Let me borrow your marker. I'll write on myself. No,
Oh.
see. 3D. If this is, oh, you're not going to see that. Anybody have? I'll just use my black. You see that's a 5 and a 3, right? And so then on this side, I'm going to need a 3 and a 5 put on there. Geez, what I do for my students. Okay? So, you see? I've got 5' end here. Can you see that? And 3 down here, and 3 up here, and 5 down here. Now, when I separate out, if I can only replicate in my 5' to 3' direction, then I've got to go from down here, because this was my 5' end, I've got to replicate from here up to here. And on this side, I have to replicate from here going down to there. And if I had 2 other arms, I would put them in there. Okay? You see where I'm going with that? So when I separate it out, I'm going to replicate from here to here. But on this side, I have to go from here to here. So I'm going to be replicating in opposite directions, aren't I? And I can only add nucleotides in that 5' to 3' direction. 5' to 3' direction. Well, no, my number 5 would be down here, 5 to 3. The new growing nucleotide chain will go from 5' to 3'. The original will be 3 to 5, yeah. But the new DNA strand will replicate and can only replicate from 5' to from the 5' to 3' direction. Now, here's something that happens when DNA replicates. It starts out at an origin of replication. Say my elbows is my origin of replication, and I'm going to separate it out, okay? And I'm going to replicate from here up to here, and I'm going to replicate from here down to here. Well, because this was my origin of replication, that bubble is going to get bigger and bigger and bigger, isn't it? Okay? So as I get bigger, say I open up like this strand that came up to my wrist will keep going, won't it? And keep going and going and going. Well, what about the other side? I have to start here and now come down to here, don't I? And then as it gets larger, I'll start up here and come to that point. You see where I'm going? Because I can only go from 5' to 3', no matter how much bigger my bubble gets, I have to start and come to that next point. Does that make sense? Okay? So what I'm doing is now I'm creating fragments, aren't I? I'm creating pieces of DNA. This one, shoo, just going. Go, 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 go. This one, I'm getting pieces of DNA as I do it. Okay? So this is a problem that ends up happening with DNA when it's replicating, is that it creates these pieces. So let's look here. Here's our parent strand of DNA. Here's our 3' to 5' and 5' to 3'. Okay? You can see how these dark blue sequences are running in opposite directions. If this was my origin of replication, my elbows, right here, coming on this 3 to 5' strand, okay, I can replicate in the 5 to 3' direction, and I'm just going to come, attach nucleotide after nucleotide after nucleotide after nucleotide. And it will just continuously keep going. This is known as the continuous strand or the leading strand. The continuous strand or the leading strand, meaning it will go and replicate uninterrupted. It'll just keep replicating uninterrupted. However, on this side, I replicated from here to here, and now I did from my tips of my fingers to my wrists, and then from up in space down to the tips of my fingers, and so on and so on. I can only replicate in little pieces, in little fragments. And these fragments are what we call Okazaki fragments. Okazaki fragments, which are little pieces of DNA, because I can only replicate in the 5' to 3' direction. So when this DNA strand opens up some more, you know, if I'm going from here to here, it opens up some more. I'm only starting here, and then I'll start here and come to this point, and so on. Okay, I can only replicate in that 5 to 3 direction, so I get these pieces. Now here's a loop for you. If this was my origin of replication, if I was going in the other direction, that becomes the leading strand, doesn't it? And then I start to see fragments leading up to this point. Okay? Got to always have something in there, right? Okay. Any questions on this 5 to 3' orientation of how these nucleotides are attaching?
What are the other items?
That's what I'm going to get. Okay? Now, these new nucleotides are attached one right after another by an enzyme. An enzyme that attaches DNA nucleotides to create a polymer, because that's what we're doing, right? We are making a polymer of DNA. A polymer, remember, is many of the same units or similar units over and over and over, making this a very large molecule. Well, the enzyme that is attaching these DNA nucleotides, this orange right here. This enzyme is known as a DNA polymer, but it's an enzyme, so what do you need to add? ASE. Polymer ACE. ASE signifies an enzyme. DNA polymerase enzyme is the enzyme that attaches nucleotides to that growing DNA strand. It creates the polymer. It allows for that polymer to be created, to be synthesized, to be made, attaching one nucleotide after the next. DNA polymerase enzyme. Okay? Now the thing is, DNA polymerase cannot join together these two Okazaki fragments though. It can't fuse them together because, quickly go back to this, it's a phosphate group up there on the top, on that 5' end. There aren't the other two phosphates. And if there aren't the other two phosphates, I can't have that reaction. So there is another enzyme that comes in and joins the two fragments together, that joins those Okazaki fragments together, and it's known as a ligase, or DNA ligase. If you ligate something, you're fusing it together, aren't you? Ligation. Tubal ligation. A female goes through tubal ligation to fuse off her fallopian tubes. Okay? DNA ligase is fusing together these fragments of DNA, these two strands of DNA, joining them together. So now, even though I had Okazaki fragments to begin with, I'm going to have one long continuous strand of DNA after the ligase enzyme comes in. Okay? Questions so far? Still not done. Life would be great if that's all that had to happen. But DNA polymerase can't just start attaching nucleotides all by itself. It needs something to stick to. Did you ever paint your walls? Ever paint your walls at home or something? What do you usually add before you start to paint? A primer. Why? Helps the paint stick to the wall or stick to the furniture or stick to whatever it is that you're doing. Helps prevent flaking and chipping and things like that, right? Well, DNA polymerase can't just start attaching. It needs something to stick to. And so there is a primer that is placed down beforehand. And this primer, believe it or not, is actually an RNA primer. It's RNA nucleotides. There is an enzyme known as the primase enzyme. The primase enzyme will lay down an RNA primer. It'll put down 10, 20, maybe 30 RNA nucleotides, making this, at this origin of replication, making this point sticky. Okay? It puts down an RNA primer. Now, what are the differences between DNA and RNA? DNA has thymine, RNA has uracil. What else? There are three major differences between DNA and RNA. The sugars. RNA is a ribose sugar, that number 5 or that number 2 sugar, number 2 carbon has a hydroxyl group, whereas in DNA it's deoxygenated. It's missing that oxygen. It just has the hydrogen. RNA has uracil. RNA is a ribose sugar instead of a deoxygenated ribose sugar. And RNA is single-stranded versus DNA being double-stranded. And because RNA is single-stranded, it's not going to want to stay on there anyway. It wants to fall off. And so we see this RNA primer laid down. A primase enzyme at that origin of replication fuses to the DNA molecule and starts attaching RNA nucleotides. Once it gets a sequence, a certain amount of RNA nucleotides, then the DNA polymerase can come in and start doing its job, attaching DNA nucleotides. This DNA polymerase also will flick off all of those RNA nucleotides and replace it with DNA. Okay? It's an opening. It's a substitute. It's a way to get it started. We've got to get that RNA. It's the opening act. You know, we got to get that RNA out there, put it on the DNA before the DNA can start attaching. Once the DNA starts attaching, it says, all right, your work is done, and it'll kick it off. The DNA polymerase will replace the primer with DNA nucleotides so that we have a completed DNA daughter strand and no RNA present. Okay? The primer starts off replication. Okay, so this starts. This attaches nucleotides, and this fuses them together. Fuses fragments. Okay? So, if we look at it all together, you can see here we've got this origin of replication, where it bubbles. At the origin of replication, the DNA is going to travel in both directions. But if I'm traveling to the left, I'm going to see one strand is going to be a leading strand and one is going to have fragments. And if I travel to the right, one strand is going to be a leading strand and one strand is going to have fragments. So you need to keep that in mind because it only replicates in that 5' to 3' direction. And if this is 5' to 3', I've got fragment, 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 leading. And if this is 5' to 3', I've got fragment, 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 leading strand. Okay? So you can see how it's going to go in the opposite direction. So overall, we've got this DNA polymerase that is attaching all of these nucleotides, one right after the other. We have the RNA primer that's being laid down by the primase enzyme to start every fragment, every Okazaki fragment has to have a primer. Every Okazaki fragment has to be started off with this primase enzyme to get that primer down there, to get that DNA polymerase in there. Then those primers are replaced by DNA polymerase also, and this DNA ligase fuses it all together. So this lagging strand, or this discontinuous strand, ends up being one complete strand after all these other pieces are done, after everything's completed. Okay? Now, there are two other molecules, two other proteins involved with this DNA replication. First of all, we have to have an enzyme or a protein that allows this DNA to unwind in the first place and unzip. Because remember, DNA is a twisting ladder. We've got to get it untwisted, and we've got to break those hydrogen bonds. We've got to unzip it. There's an enzyme known as a helicase. Is that on there? Oh, it's right here. This is our helicase enzyme. Helix, once again, with ASE, saying enzyme. Helicase enzyme actually will unwind and unzip that DNA molecule. But now those hydrogen bonds are going to want to come back together because it's that attraction. And so we have this helicase enzyme unwinds and unzips. Once it gets it unzipped, other proteins come in and bind too, which are known as single-stranded binding proteins. Single-stranded binding proteins. And what these will do is keep it as a single strand. They'll bind on both sides, these proteins right here and up here. They are binding at that fork. They are kind of moving down. As this DNA strand just continues to open, those binding proteins are moving along with that replication fork as it pushes it apart. The helicase enzyme is up ahead. unwinding and unzipping. These proteins are keeping it separate, keeping it as two individual strands, allowing the daughter DNA to form all the way along it. Does that make sense? Okay.
So the overall direction of replication for the condition called what is it, continuous?
The continuous strand, well, I mean, it just depends. This is a continuous strand, and so it's going to open up this replication fork in this direction. Okay? The direction of the continuous strand is what's going to allow that DNA molecule to keep unwinding and unzipping. Exactly. Because in this direction now, the bottom strand that is fragmented here will become the continuous strand going to the right. And because it's going still in that 5' to 3' direction, it'll just keep on going. And it'll open up the fork in that direction. And that's why eventually the two pieces will come together. When you have that. Because now as they're pushing, say this is our leading strand here, and this is our lagging strand on the top here, eventually they're going to come together. And I guess we should do the leading strand on the bottom, because that's how our picture down here shows. The leading strand on the bottom and leading strand on the top going in this direction. But eventually those forks will find each other to where we end up with two completely identical daughter cells. So now during synthesis, when DNA replicates, and we were showing DNA replicating, making identical daughter cells, and how during mitosis those identical daughter cells separate, that's what we're talking about. How this complementary base pairing is so exact. And in attaching those nucleotides, DNA polymerase may mismatch a nucleotide. Maybe 1 out of every 100,000 nucleotides might be mismatched. And maybe 1 out of every million might be forgotten. Because DNA polymerase also acts as a proofreader, that spell check, that, oh, you're not spelled correctly, get out of here. You know, that little red squiggly line that comes underneath your Word documents in the computers anymore, saying that it's spelled wrong. Well, DNA sees that and spells it right, puts the right nucleotide where it's supposed to be. I'm sorry, DNA polymerase sees that. It's that proofreader, making sure everything's the way it needs to be. Sometimes it does screw up. When you're writing a long report and you've been looking at it for days and you're reading the same thing. You're kind of like just scanning it over. You're not catching the little details. That's why you give it to somebody else to proofread, right? You're like, look this over, make sure it sounds right. Don't you usually do that? And then somebody is going to catch the TOW instead of TWO. Your spell check didn't catch it. You read it 100,000 times and you didn't catch it because, you know what it's supposed to say. The human mind is an amazing thing. I can write up here, the human mind functions in a part that it doesn't matter which order the letters are placed, as long as you know the letters of the words are in the right order. This is great language. Great. Can you read that? Dead boys know not. It does not matter anything. I have spelled absolutely nothing correct. But the human mind, as long as the first and last letter are in the position where they need to be, if it's the first and last letter, you knew that said dead boys. Even though the O and Y were switched. I mean, as long as the first and last letter are in the right position, whatever happens in the middle doesn't mean a thing. You can still read it because of the way the human mind works. And so when you're seeing something like this, well, it might catch it, but you might not. Your spell check's not going to catch it. DNA polymerase would go back and catch it. If I was proofreading your paper, I'd go back and I'm like, you meant to say to, not toe. Right? I would catch it. type of thing, because I'm an editor, I'm a proofreader, I'm going back over it a second time. DNA polymerase will very rarely, very, very rarely miss a mismatched nucleotide. Mutation. That's it. A mutation is a mismatched nucleotide. Quite often in this long strand of DNA or whatever that we have here, out of the 6 million nucleotides we have, only about 300,000, we only have about 300,000 genes. And how many nucleotides those genes are, you know, probably range anywhere from 10 to, I don't even know if there are 100,000 nucleotides in length type of thing. So we're not taking up those 6 million pieces of DNA, those 6 million nucleotides of DNA. A lot of it is junk DNA. If a mutation happens, it doesn't matter. If it's a mutation not in a gene, if that DNA is not mismatched in a gene, you'll never see the side effects of it. But as soon as it's inside the middle of a gene, you got sickle cell, or you've got, you know, whatever. You've got that lactose intolerant. You've got malfunctioning proteins, or whatever. Lactose intolerant is a mutation in the DNA that doesn't allow you to make the protein that help digest milk products, dairy products. It's a mutation in the DNA. Male pattern baldness, it's a mutation in the DNA. Okay? So, but DNA will replicate almost exactly every time. Now, when I draw out the DNA like this, when we were talking mitosis, each strand is a strand of DNA. Each strand is a double helix. When we're putting the two together like this, imagine your centromere kind of like right there between the two strands. Okay? That's when we're replicating it during the synthesis phase of interphase. That's what's happening, is we're making these exact copies of daughters. And you see now how they are exact copies of DNA. Those complementary base pairing, A to T and G to C, every time. Okay? Got a light bulb? Oh, turn it on. Chromatids are DNA. These are sister chromatids. They're identical strands of DNA held together by the centromere until anaphase, or anaphase 2. Got to throw that one in there too. Any questions? This is just a little chart here that describes these different enzymes that we've been talking about. That's all. You got your helicase and single-stranded binding enzymes that help unwind and keep the DNA unwound and separated. The primase enzyme is there for priming, adding the RNA primer to it. DNA polymerase is for elongation, replacement of the primer, also the proofreader. In the lagging strands, you also have Okazaki fragments, so you have the ligase enzyme that helps join them all together. Just puts it all in a little chart there. Everything that I had right here, then mumbo jumbo. Okay. Any questions on DNA replication? Or on any of the scientists? The last little section, she just turned her page. There's a little section talking about telomerases. Not going to go into it. Okay. Telomerases is there's a problem when it comes to the end of replicating the very end pieces of DNA, because where do you put the primer, or how do you get the DNA? Telomerase enzymes are this function that creates this replication, whatever. It like makes this little loop and all that stuff. I'm not going to go into that. So.